Hi everyone, and welcome to second day of the 8th International Meeting on Brand Building. My name is Israel Soares. I will be sharing this session, and we will start today with a brilliant speaker in quantitative genetics, biometrics, and statistics, Julian Garcia. Julian holds a bachelor's degree in biotechnology and a master's degree in computer science, artificial intelligence. He is PhD student in data analysis at the Center for Biotechnology and Proteomics at the Polytechnic University of Madrid and is resident in artificial intelligence plant biology at Google X. Today, Julian represents on integration of multiomics and the interactions in product modes. Please welcome Julian.
So first of all, I want to thank the organization because they are the best hosts that I have ever seen. Thank you guys, you are doing great. And secondly, for giving me the opportunity to share this spot with such a great of amazing researchers. I truly feel as a fish out of the water. So yeah, I'm Julian Garcia Vadillo. I'm a PhD student at the Universidad Politécnica de Madrid. I study quantitative genetics and plant breeding. But the last year I have been in the US, first as a visitor scholar at the University of Florida under the supervision of Dr. Diego Jarquin. And after that, I've been an AI resident at Google X. I can't really talk about what I did there, but today we are going to discuss two applications that I did during my time in Florida that gave me the knowledge and the skills to pass the interview, so I guess it's also interesting. Okay, so let's go. First of all, this is something that Diego likes to ask their qualifying students. What's an interaction? It's something that we are always saying, but if we have to define it, that's interesting. So I like to define an interaction as a situation in which one variable, the effect of one variable on outcome or a variable that we want to target depends on a third variable. And this happens because the relation between the variable and the outcome is not linear, it's not additive. We need a third variable interacting with it. So in the context of plant breeding, we have two important examples of interaction. One of it is the G by E, the genotype by environment interaction, and the other is the specific combinability when we talk about modeling hybrids. So in the first equation, we could say that the phenotype of the environment AI and the genotype J depends on the environment plus the genotype plus the G by E interaction. And the same way, the genetic value of a hybrid depends on the general combinability of one of the parents, the general combinability of the other, but also the interaction, the SCA, which usually models the dominance. And why are interactions important? Well, yesterday we had two talks that referred to G by E. The first one was Fernando, that he was trying to deliver, his company was trying to deliver a variety that is well adapted to each of the environments or the cluster of environments or target population of environments. So they were looking for lines that are superior in that specific genotype. On the other hand, Alex talked about growing corn in South Carolina, which is outside the Corn Belt. For that, he referred as selecting against G by E. I prefer to call it select for stability, meaning that we want to reduce that G by E term, but we want a line to behave similarly in all the environments, more than half an environment in which that genotype is really good, which is the superior. So yeah, G by E is interesting for the commercial value of varieties because yeah, we can improve the genetics, but what we are eating is a crop that has to be grown in the floor, in the soil. So yeah, this is how I like to see G by E. For me, G by E is just a statistical trick that allows us to model changes in rankings across environments. This is how I see it. So imagine this simple scenario. We have a high resource environment or an easy environment, and we have a low resource environment or a challenging environment. And then we have a genotype that I call racehorse that is specifically is adapted to this high resource environment, so it has a really good performance on it. But when we move it to a low resource environment, the performance drops. On the other hand, the genotype warhorse It's not the best in the high resource environment, but see how it's stable. This slope is almost zero, meaning that the variability across environments is really low, and that can be interesting if we are facing climate change or changing environments. The case of specific combinability is probably even more interesting because it allows us to model heterosis. And heterosis is, how to say it? Do you use Excel, Microsoft Office? You have to buy the license, right? Like you have it in your computer, and if you want to put it in another computer, you have to pay it again. You can copy it. This is something similar. Institutions or companies that have the parents are able to sell the growers hybrids, F1 hybrids, that are optimized for that specific combinability. But growers are not able to reproduce that because the seeds of their progeny, the F2, has already segregated. So it doesn't have the properties of SCA that the hybrid has. So it's a way for companies to keep the software private. Or it's a really good business model. This is why heterosis is that important. Okay. And now, let's talk about omics. I like to define omics just as a data set that characterizes a biological factor that is interesting for a research question. So if we are working with plant breeding, we have environments, we have genotypes, we have treatments, we have management, we have a lot of things, and each of them are factors that we can characterize with the appropriate features. So in plant breeding, we have genomic markers that are usually characterized in genotypes. We can also have pedigree or any other kind of information. We have weather covariates or soil covariates that will characterize environments. And we have phenomics, transcriptomics, or other omics that, since are extracted from a plant in a specific moment, are characterizing both the genotype and the environment at the same time. And I like omics because they allow us to borrow information, which is the core of AI, is the core of machine learning. Is we can predict things that we haven't seen because we can resemble them to things that we have already seen. That's the power of borrowing information, and that's the core of genomic selection. Okay, so let's talk about borrowing information. Imagine this marker matrix. I don't know if you are familiar with molecular breeding, but this will be a data set for genomic selection. We have some genotypes and some markers. And in this case, the similarity matrix tells us that genotype 1 and genotype 2 are kind of close, genetically talking, while genotype 3 is kind of unrelated. So now, if we go to the phenotype, we see that genotype 1 is big, it's a high-yielding plant, while genotype 3 is a low-yielding plant. So can you tell me how it's going to be genotype 2? Well, we could think that it has to be a big plant, right? But that's not really true. It depends on the heritability. Heritability tells us how much of the phenotypes that we see can be imputed to the new lines just by relatives. If this trait it has a low heritability, even if the relatives are good, given that the phenotype depends more on the residual or the environment than the genetics, it's not really useful. So it's important to have in mind heritability as the proportion of variance, of phenotypic variance that is explained by the genetics, and in a more general setup, any omic, the variance that any omic explains respect to the phenotype. That's a way that's a measure that we have for assessing its importance. So we can do the same also with environments. Here we have I gathered data from three cities, Gainesville, Piracicaba, and Madrid. And I gathered some environmental covariates. We have latitude, longitude, temperatures, and rainfall. So they are really different. We can construct this similarity matrix, and we could eventually use this for improving our predictions. But it's yet to see if these variables are actually good to predict what we want to predict. We can also characterize more than one factor at the same time. As I have said, when we use phenomics or transcriptomics, something that needs to be collected as a sample from the plant, we have an interaction, already an interaction. For example, this indexes and DBI is collected from the RGB or the multi or the hyperspectral bands. And that's collected at the plot level from a plant. So it's both an environment and a genotype. And using this, we can characterize the relation between the genotype by environment factor. Not genotype, not environment, but the interaction. So in this case, for example, we have a cluster which is involving environment 1 and another involving environment 2. This means that in this case, environment is being more important in terms of characterizing or grouping levels of our factor. Okay, now we are going to see two applications. This is the first. This is not involving omics, just G by E. And the idea here is to leverage G by E to reduce the phenotyping efforts. And it's called a sparse test. Okay, so in breeding programs, as in life, we have problems, we have goals, and we have limited resources that we want to optimize, and we want to do the best with what we have. So in the context of breeding programs, we can have low budget, we can have economic constraints, we can have space problems, we can have a seed lack, problems with seeds. And in that case, we need to think about how to allocate the resources in an efficient way, right? So when we are exploring a huge space of solutions that we can really handle, we can use heuristics. This is a couple of heuristics that are kind of two strategies that are kind of opposed, that are explore or exploit. In the case of explore, in this context, it will be more like, okay, if I have limited spots, I prefer to test as many genotypes as possible, so I can improve my yi in the genetic equation, the intensity of selection, and I can see the variability. Or given that I have some kind of already good genotype, I would like to exploit it, meaning that I will evaluate it in many environments to see if it's really a racehorse, a warhorse, or a line that is not worth to keep in my program. Yeah, you could argue that, yeah, this is how a breeding program works, right? The first stage or the first phases are more like exploring, like we have many different lines that are tested only once or a few times. And then once we have some promising lines, we advance them and we try to test them in multi-environment trials. But in general, even in the last phases, we might have more lines that we want to test than actual space. So yeah, imagine this problem in which we have just 3 genotypes and 3 environments, but we have a budget for just 3 phenotypes. Phenotype is the combination of a genotype in an environment. So imagine that we can plant only 1 genotype per environment. So which combination is going to give us the most valuable information? We can explore, meaning that these are the environments, these are the genotypes, and yep, we can say, okay, let's try in each, since I only have 1 spot in each environment, let's try each genotype so we can see all of them. That will be the exploration or the explore strategy. And we can exploit. Let's go with genotype 2. Let's get it in the 3 environments to see how it behaves. Let's see if it's stable, or if it's superior, or if it's not a good line. So, when we ask this question, what's the most valuable information, we have to ask also, valuable for whom? And in this case, is for our model. So our idea here is to, given that we have limited resources, let's use that phenotypes to train a model, a genomic selection model, to predict the rest of the grid. So we can see or have a proxy of all the genotypes in all the environments. That will be the idea. Okay, so we did this in a sugarcane breeding program in Colombia. I'm not an expert of sugarcane as many of you, so please be kind with the questions. So we had 6 environments and 186 genotypes. We had all the phenotypes, the complex picture, which is 1,116 phenotypes. And we also had genomics. We had an array of more than 22,000 SNP markers. And we measured 2 traits that are the components of what was discussed yesterday. Yesterday we were talking about sugarcane per hectare. This is the 2 components, the ratio of sugar and the raw weight. And the framework is more like, let's use just 1 seed. Like in the previous example, let's assume that I can plant only 31, which is 186 divided by 6, I have only 31 spaces in each environment. That's all I have. And then I have to allocate those 186 genotypes. So what we are going to do is to randomly assign them either to the group non-overlapping genotype or overlapping genotype. If a genotype is non-overlapping, then we are going to see it just once in one specific environment. Genotype 1 is only going to be seen in environment 2, that's all. But if the line is overlapping, we will treat it as a check. We are going to evaluate it in all the 6 environments. So in order to have an overlapping genotype, we have to remove 5 non-overlapping genotypes that we won't test at all because of the constraints. Okay, and this is a way to see the allocation design. So we label each strategy with 2 numbers, A and B, representing the number of non-overlapping genotypes and overlapping genotypes. So in one extreme, we have the 31, 0, which means that we are going to test 31 different genotypes in each environment. And we have it here. So this is, the columns are environments, and the rows are groups of 31 genotypes. So you can see how we don't repeat genotypes across environments. If we squeeze these columns, we will see that we are testing once every genotype. So we see everything, but only in one environment. In an intermediate strategy like 16-15, we have kind of a mixed thing. We have 16 genotypes that are going to be unique for each environment, but then we are going to have 15 common checks. This is basically an augmented design with a lot of checks. And we are varying from 28.3 to 427. 27 checks is a lot. Oh, sorry. And finally, we have the other extreme in which we have 0 non-overlapping genotypes and 31 overlapping genotypes, meaning that we are testing 31 genotypes as checks. So this is really good for characterizing G by E because we are seeing a lot of lines many times, but we are removing 5 seeds of the genotypes. We are not seeing them. We have to predict them. See how here, while here we are seeing all the environments, if we squeeze this here in this other stream, all these lines are not being seen in any environment. So this last, if you are familiar with genomic selection, this last scenario is similar to a CV1 cross-validation scheme in which we predict genotypes in already seen environments, but genotypes that haven't been tested before. Okay, and these are the models. Models here are not that important. The important part here is how we build the train and the test sets, but actually you could use any model. We like to use mixed models, linear regressions, and Bayesian frameworks, but the important part here is how to build this and test your own models if you want to try it. So our models are 3. The first one is the E plus L is the environment, the main effect of the environment, and L is the main effect of the genotype. But we use L because here we are not introducing genomic information. So it's more like lines are independent. Genotype 1 and genotype 2 could be called Julian and Pepe. It doesn't matter. They have no relation. In the second model, we add the G, which is the genetic effect, but for the genomic markers. In this case, the genotypes are characterized by a kinship relationship. Just as in genomic selection, we have the markers that are telling us that genotype 1 and genotype 2 are no longer labels, but they are siblings or relatives or whatever. And in the third model, we introduce the G by E interaction, which allows us to model the multiplicative interaction between the genomic markers and the environment. Okay, and why do we need these models? Well, the first one is kind of a baseline. It's not a really good model. Actually, it's just like taking the mean of the environments, taking the mean of the genotypes, and assuming that that's going to be the prediction. So this is a really limited model, and actually in scenarios like the 031, like the CV1-like scenarios, it's totally useless because given that we are not seeing genotype 2, we can't do anything to predict it because we don't have information to borrow. Is that clear? In the second model, that changes. We have the markers. This is a genomic selection task in which we can predict new genotypes that we haven't tested because we can borrow information between them. And the third model here is the key, is the one that allows us to model not only the prediction of new genotypes, but also model changes in the rank. This allows us to model a new reality in which we have changes in rankings across environments. And here we can see this with an example. This is simulated data. So imagine a world without G by E. This is how a world without G by E looks like. We have environments, environment 1, 2, and 3. You can see that the environment 1 and the environment 3 are kind of better, favorable environments with a higher mean. And we have 4 genotypes. But see how the lines are parallel, meaning that the genotypes are behaving the same. So genotype 4 is the best in all the environments. There's no selection here. You will just go with genotype 4. So according to this, if we compute the genetic correlation between the environments, we get a 1 because the rankings are the same. It's like there's no selection here. This problem becomes trivial because once you have seen a line once in one environment, you know how it's going to behave in the others because the rankings are not going to change. Then why bother? But reality is more like this. We have crosses. We have changes in rankings. Here, for example, we could define genotype 2 as a stable line that behaves similarly in all the environments, while genotype 4 is kind of a superior line. It's a racehorse. It's really good in environment 1 and environment 3, but it's not good in environment 2. And if we compute the genetic correlation now, we have something like this. See how the lines that are good in environment 1 are also good in environment 3. This is why we have a high correlation here, but in the other case, the correlation is negative because the rankings are totally swapped. This is the reality in our data. We have the 2 traits here. This lower matrix, triangle matrix, is the sugar accumulation, and this one is the raw weight. We have correlation that are positive, but they are changing. These values means that there is a lot of changes in rankings. The lines that are good in one environment are not necessarily good in others. So this is why we need G by E, basically. Okay, so we repeated this allocation strategy 10 times, each one randomizing the lines that are checks and the lines that are non-overlapping genotypes. And we compute the average. Well, first we use 2 metrics. We use Pearson correlation to account for the rankings, but we use mean square error to also account for the actual value. So we can have really good predictions in terms of accuracy if we are able to predict the rankings. But if we want to approximate the actual value of the phenotype, we need this error to measure that metric. So in terms of accuracy of correlation, we can see that as we reduce the number of non-overlapping genotypes and we increase the checks, the accuracy drops. That's really interesting. Like, we expected that for the gray model because remember, we don't have anything to relate genotypes. So if we remove genotypes from the training set, we are not able to predict. So we expect this to drop to 0 eventually when we have 0 lines non-overlapping. But you can see that models the model 2 and model 3, the G and the G by E, are behaving similarly in terms of accuracy. But when we go to the mean square error, we can see that specifically in the scenarios with more non-overlapping lines, the orange model, which is the G by E, is behaving a bit better. Yeah, so think that here we're using only 1/6 of the data set to train the models. Usually when we run 5-fold cross validation, we're using 4 folds for training, like 80% of the data set, and only 20% for testing. Here is more like the opposite. It's a 6-fold cross validation in which I'm using 5 as a test and only 1 as a train. So I think this is pretty good. And we even reduce a bit more the training sample size. We started with 186, which is 1/6, and we reduce it until this limit, which is 72 phenotypes. So we would expect the accuracy to decrease because we are reducing the sample size, but we found that, for example, here for sugar, we have some local maxima here. This is really interesting. This 12-0, meaning 12 non-overlapping lines, is behaving, is predicting better than, I don't know, 16-14, like scenarios with more data. And this is across 10 replicates, so there must be something there. We are not sure. It might be population structure. It might be something that, I don't know. This is really complex. Okay. So the conclusions of this study were basically that the more overlapping genotypes, the more checks that you introduce in your training set, the better the accuracy. No, sorry, the worse the accuracy. So we want to have many lines, even if we can see them only once. Also, we will have expected the GBI model to behave a bit better, but it's not the same to predict just one seed than compute the variance components with all the data sets. So there are many things that we can do, and theory is not always like what we should follow. Yeah, another interesting thing is that the reduced sample size got a better accuracy, which was not expected. In fact, some studies, similar studies that have been conducted previously, this one in wheat and the other in soybean, you can see how the model one, the one which doesn't have the markers, always drop to 0 because of that inability to relate or to borrow information. But both the G model and the GBI model are kind of stable, and they improve a bit when we reduce the number of non-overlapping lines, when we increase the number of checks. So we think that this is because our dataset, sugarcane dataset, was kind of small, but yeah, we would like to try a bigger dataset to see if that trend, that decaying trend is maintained, or we can actually revert the trend. And in this study, we just use markers to relate lines, but if you have environmental covariates, it will be possible to have a E term, which is just the environment, but also a W, which is kind of the kinship matrix for environments, to try to improve a bit more the model by giving context about the environments. And yeah, this study was published some months ago, so if you want to check it, it's that one. Okay, wow. Okay, now, let's talk now a bit about integration of multiomics. The previous study had just genomics, and here we have everything but genomics. We are going to try to find surrogates for genomics in the world of phenomics. So, as you know, these days we have the ability to collect many different data sets with different nature, and sometimes we don't even know what to do with them. Many people have asked me what to do with this specific information that I collected for a different thing that it might be interesting to introduce in this other hypothesis. So the introduction is far from trivial. One option that we have is to use these AI methods, like for example, random forest, deep learning models in which you kind of just take the data, stack it, you have, I don't know, markers, you have environmental covariates, you have soil covariates. You put that together in a vector and you just fit the model. Random forest, give me the predictions. Deep learning, give me the predictions. That's possible, but I don't really like it because that's not easy to interpret. Like it's really hard to model things inside. Those models are kind of black box. You put the data, you get your output, but you are not really telling the model where do you plan to find interaction, or you can ask the model about your hypothesis. This is the way I see it. So our approach is called the reaction norm. This was a model developed by Diego in 2014 and collaborators. And this reaction norm model was designed to deal with the problem of GBI. So in that specific data set, they had genomic information, but also they have environmental information. And see how, as they increase the complexity of the model, introducing environmental covariates, they reduce the residual variance. They are stealing, we have a residual term that is like, we don't know what is there, but as you use more information, you are able to steal information for you. It's like, okay, now I know that this residual is due to this omic. That's really important. So we are using this approach. In this case, we don't have genotypic information, but the concept is similar. And here is my favorite slide of the day. Let's try to understand a bit how matrices work for this. So let's say that we have 3 genotypes, and we have only one environment, but we have 5 spots, and we want to place, I don't know, genotype 1, genotype 3, genotype 2, whatever, more than one time, okay? At the level of the genotypic level, we have only 3 entities, or 3 levels for the factor of genotype. We have 3 environments, that's N sub G, and we have P sub G markers, that can be 1,000, 20,000, whatever. So if we multiply this matrix by its transpose, we get this nice genomic kernel, which is the G matrix, the Van Raden, whatever. You can call it whatever you want. This is the G matrix that we use in genomic selection. And it has the dimensions of the number of levels, which is 3 here. And it's basically telling us what's the relation between one genotype and the others. On the other hand, we can have a plot level omic. Imagine that we are collecting a sample of the soil for each of the 5 spots. That's a totally independent source of information. It has nothing to do with the genotypes. It has to do with how is the soil. So we can have, in this case, 5 rows because each spot has its own information, and we have P sub O variables. It can be, I don't know, nutrients, it can be humidity, temperature at 1 centimeter, whatever. And the process is the same. We multiply that matrix by its transpose, and we obtain what we can call an omic kernel, which is a variance-covariance structure. The problem here is that, as you may have noticed, they don't have the same dimension. This is 3 and this is 5, and that's because every level has its own specifications. You have these levels, you have these variables. This is the problem with omics. But we can do a trick here with this 3x3 genomic kernel, and using some design matrices. These are binary matrices telling us what's the relation between the 2 variables. So basically, this is a 5x3 telling us that in the first spot, I have a genotype I have the genotype 1. In the second spot, I have the genotype 3, and so on. If we multiply this Z matrix by the genomic kernel, by the transpose of this Z matrix, we get an extended genomic kernel that now has the same dimension as the omic kernel. That's because we are relating both data sets. And if we, now, this is the hard part now, that they have the same dimension, we can multiply them, well, okay. This is a matrix multiplication. This is just a Hadamard product, which is element-wise multiplication. So we need the matrices to have the same dimension for that. And we can get this nice G by O interaction kernel. This kernel is going to tell us about the relation between the 2 variables, and is the way to model interaction with omics. So keep in mind these 3 terms, these 3 omic kernel, extended genomic kernel, and GO interaction kernel, because now it's time to build the linear predictor. You have seen this in many papers. You have a nice equation telling us, okay, my phenotype is going to depend on an overall mean, a genotype effect, an omic effect, a G by O interaction effect, and a residual. And what's the point of the kernels? Well, all of our terms except the mean. The mean is a fixed effect, but the rest of them are random effects that follows a normal distribution with null mean and a variance covariance structure, which is proportional to the kernels that we have construct times a scaling factor. These scaling factors are the variance components, and those are key for us because they allow us to know what's the importance of each omic in our outcome. So if we remove the omic and we keep just the genotype, we have the GBLAB model. And this ratio between variant components is just the heritability. Genetic variance divided by the sum of genetic plus the residual. So basically, if we remove from the numerator the G and we put the O, we get the pseudo heritability of that specific omic for that specific trait. Okay, enough math. So now we are going to apply this to Pine Loblobby tree problem. They were having problems with cone abortion. They were having a lot of conelet abortion. And the problem with this trait is that it's really time-consuming and error-prone because you have to count the conelets twice, like 200, 300 conelets per tree, first in the spring, and then 3 months later at the end of the summer, and then you do the operation to get the percentage of abortion. So our idea here is to use. So our idea here is to use this this omics as well. Abortion rate is kind of telling us about the health of the tree. So we are trying to use some omics as a proxy of the health of the tree. So basically what we did here is let's collect nutrients from the leaves and let's run drone flights 2 times. So we ran a multispectral camera in a drone in spring before the first count of conelets and in fall after the second count of conelets. So we get this R, which is the ratio, is the abortion rate, and we got these 3 omics: nutrients, Phenomics or multispectral for spring, and phenomics or multispectral for fall. Yeah, and now this is important. This is not genomic selection. I want to remark that here we are in an orchard. We are trying to predict the phenotypes, not the breeding values. We are not using these trees as a partners for the next generation. We are just trying to reduce the phenotyping costs. So in this case, management is more important than genetics. And then, yeah, as I said, we want to predict the phenotype. And even if we want to have breeding values, we can't, because for breeding values, we need genomics. The things that we are capturing when you collect the nutrients, when you collect the phenomics, those are expressions, those are complex functions that involve genetic factors, but also non-genetic factors. We have environment, we have management. Even inside the genetic factors, we have the additive part, but we have also the non-additive part. We have the dominance and we have the epistasis. And this is a really big problem with omics, that they can never substitute genomic selection because markers are the only phenotypes that have an heritability of 1. That's the only thing that we have there, and we can say, this is the marker, this is going to be there forever in every seed. The moment that we use omics, even if it's a NIRS in a seed, we have other effects that are not going to be inherited. So we have to be careful with that when we are using them as breeding values, or we're using those predictions as indexes for selecting parents or move genotypes to the next generation. But for us, here it's okay because we want to predict phenotypes. Yeah, sorry for that. Okay, so here's the data set. We had 600 trees planted in 3 orchards over 3 times. So we have like 9 environments. And as I have said, cones are counted twice in June and August, and the abortion rate is a relative loss of cones. And for each tree, we have 3 kernels. We have as the omic kernel, we have 3 data sets that are, imagine, the spring is all the trees in the rows, and then all the multispectral bands in the rows. Those are our features. The same for the fall. And in the case of nutrients, we have the same. All the trees, and then we have calcium, ratio between sodium and potassium, things like that. Those are our variables. So we develop a predictive pipeline with 2 goals. The first one is to determine what's the usefulness of each variable. Are omics useful? And even within omics, is the ratio between potassium and sodium useful? That's the scientific question. And then we have the application, which is let's develop methods that are able to predict the phenotypes without having to count the conolets. And we ran a lot of models, a complex framework under this Bayesian mixed model using BGLR. Okay, now we have to talk about metrics. I have 40 minutes, okay. So here we have the 3 cross-validation schemes that we use to assess the models. If you are familiar with genomic selection, you have probably heard about them. We have CV2, which is just sparse testing. We are just removing, this is 5-fold, sorry. We are removing 20% of the lines, of the genotypes, just by random, meaning that we can predict lines that we have already seen and environments that we have already seen. In the case of CV1, we have some constraints. In this case, we are removing all the observations of one line or many lines from all the environments. So in this case, we are predicting genotype 2 in all the environments. We don't have any information about genotype 2. That's important. And in the other case, in CV0, is the opposite. We remove all the information about one environment. So we are trying to predict a new environment. And this is really useful if we are predicting a new year. So we want to evaluate a model to predict a new year of whatever thing, because that's a totally new environment. And regarding the metrics, we are using again both Pearson correlation and mean squared error. But now I want to remark that we are using Pearson correlation within environment. So we don't compute a single correlation between predicted and observed. Rather, we first compute the correlation within environment, and then we average that. And I think this is important because here, this is synthetic data in which I simulate 2 random clouds of points, but I gave the second group, you can think about this as environments, I gave the second group a mean of 2. So this cloud of points is centered at 0, but this other cloud is centered at 2. And see what happens. The correlation is really good. It's 0.63, you have a really low p-value, everything seems to be perfect, your model is good, you are going to be promoted. What's the problem? The problem is that this is a spurious correlation. If you do the within environment correlation, you will see that your correlation is 0 because you have nothing inside the environment. And that's an artifact called the Simpson paradox that affects a lot of models. I have seen this many times. Correlations that are computed across without taking into account that there is a third variable, in this case the environment, which is inflating our accuracy. So basically this model, a model that does this is useless. We don't have any information about what's going on inside a specific environment. We are just good at predicting the average value of each environment, but that's not really useful. Okay, these are the results. So this is the initial correlate number, the final correlate number, and the abortion rate. Its color is a model. Yeah, this is the correlation, and this is the mean square error. We are going to talk about the correlation. So the three models that are kind of the three models that get some accuracy in CV2 and CV0 are the ones involving the G. So we have G, omics plus G, and omics plus G plus all the interaction. That's the dark model. That means that the only thing that is actually giving us information is the genotype. We don't have markers, but we have many reps of the same genotype across the ORCADs. This means that if we are able to see genotype, a clone, we have enough information to predict the others. But omics are not really helping us. We know that omics are not helping us first because when we use them alone, the accuracy is really low or even negative. And also because see what happens in CV1. In CV1, we are removing all the observations for a set of genotypes. If you remember the first model I showed for sugarcane, the model 1 that drops to 0 in CV1, this is the same. It means that we have nothing. If we don't have the genotype, we have nothing. That means that our omics are not good enough to borrow information and to relate our factors. We have something here. It's true that the models are a bit better sometimes when we introduce the omics, but I don't know if this is worth the effort. Another way to see it is with the variance components. Here we have the relative importance of each omic for each model. So when we use the model phenotype is equal to just the nutrients, we see that the variance is less than 1%. It's explaining a really low variance. It's like heritability of 0. Only when we introduce the G term, we are getting some kind of variability explained, but it's 20%. It's not like 100%. This is why our models have a low accuracy. This is this gives me some hope. This is a orthogonal gain that we are stealing some residual. Using the omics, we are stealing this to the residual. We are telling, okay, this is explained by genetics, but then we are explaining something with the omics. But this is not worth the effort of collecting all the omics. So at least for this specific case, the omics are not really useful. I think the framework is interesting and it should be tested in other datasets, but for this specific, these two first, these kind of patterns in accuracy and these variance components are telling us that these omics, are this as we have thought about it, are not good enough. So yeah, basically those are the conclusions. We had something in CV2 and CV0, which is actually good because CV2 can be used to just do a sparse testing. Like, okay, I have 300 trees, but I just want to measure 100. Let's predict the other 200. Let's run the drone and let's predict them. That will be an application for CV2. And CV0 will be something like, okay, let's predict what's going to happen next year without even having to phenotype them. But still, genotype is the most important factor, and something that would be really nice to have is markers. If we had markers, we could first relate individuals, but then we could improve probably the interactions, like how the markers interact with the omics. We could have something like, what's the interaction between marker 1 with this specific sodium value? That will be interesting. And yeah, of course, we will improve the CV1 scenario in which we aren't able to predict anything here. This is really hard to predict for any program, but it still should be a bit better. Okay, I guess I have 4 minutes. Okay, now I want to talk a bit about Chido, that if you know some Mexican Spanish means cool, and it's a software that we have developed for this kind of applications in Diego Harkin's lab. So Chido is an open source software developed in Diego Harkin's lab by Francisco. He's a Google employee, like a real Google employee, but he's also a master student. This is built in R, it's a Shiny app, but it's running in VGLR basically. And the idea is to give access to people to run this kind of models with omics and cross-validation schemes without having to program. Like you just have your data, you kind of clean it, and then we run the models, and you don't have to run, you can just collect the results. So the idea here is a bit, we have an interface in which you will introduce a phenotypic file, which should have at least 3 columns. Should have environment column, genotype column, and phenotype. And with that, we can run what I call the model 1. We can call a model which says this phenotype depends on the environment plus the line. But of course, we want to introduce more omics. So here we can introduce genomic markers, which will give us the G term. We can introduce phenomics, we can introduce environment, we can introduce pedigree, of course, and high throughput, and almost anything that is characterizing a biological factor. The only thing that we need is to relate each of the factors with the original phenotypic file. So if we are inputting environmental markers, we need to tell, okay, this 1 here means that we're in the environment 1, that this variable depends on the environment 1. It's not like this is information from the genotype 1. This is kind of, if you know something about databases, this is foreign keys and primary keys. This is kind of relating genotypes using join operations. Okay, once we have that, we will have this screen with the available omics, and this is really nice because we can drag the terms. We have E, we have L, we have G. We drag the terms with the mouse and we put them in terms to add. Once we add it, we are building the main effects of our model. We want a model with E plus L plus G. This is model 2 from sugarcane. But the funny part is that if you put 2 terms together and you click add, then you will introduce the interaction. So we can do as many interactions as we want. We can put E plus L plus G plus A. It's going to run. I don't know if it's going to be really useful, but you can do it. After that, we will set the hyperparameter tuning because this is a Bayesian framework, so we need a number of iterations to make the model converge, and we need a bounding rate to remove the first iterations, which are kind of random. So we need to remove them to account for the random effect that we have at the start of an iteration. We can also filter markers here. We can center and standardize the data, and here we will select the model that we want to run, that we have already created here, and we will choose which CVs we want to run. CV1, CV2, CV0, I have already described them, and CV00, which means predicting new environments, new genotypes in new environments. This is the hardest thing that we can do with a multi-environment trial. This will give you some results like this. It will output a CSV file with all the results, all the variance components, everything that I have explained for the omics dataset, and also some plots. And yeah, that's pretty much all.
We have time for questions. Does anyone have any questions?
Model, because that's a decision you make, right? You cannot just include everything.
Yeah, that's a really good question. Usually when we work with, it's not me, no. When we use markers, we tend to select everything. Well, we have some kind of quality control, we remove math, but in general, we keep everything. We assume that that's useful. But in the case of environment, that's more complicated. My approach will be to try. Just try. Get a model like this, try some variables, construct your kernels, and run the models and see which variance components you can get. The higher the component, the better you are doing with your environmental covariates. I think that's a trial-error process. Yeah, also it depends because we want covariates that cluster environments in the same way as the phenotypes are clustered. So if we have, I don't know, if we have a breeding program and we have 2 different TPEs, 2 group of environments, we would like to have covariates that cluster things in the same way. Depending on the covariates, the models are going to be clustered in different sets. So another approach is, before doing all the kernel and all the modeling, just run a PCA and see how it is, or any kind of clustering algorithm, to see if the environments are clustered as you would like them to be. But this is more like trial error and a lot of running. Good question, thank you.
Okay. Julian, we would like to thank you for your lecture. We would like to offer a small gift. Okay.